So a very warm welcome to the first in this series of webinars organised by Writtle University College and Liberty Produce and sponsored by ERA. We're delighted that so many of you can join us. This is the first of 3 seminars over the next 3 months, aiming to bring together industry colleagues, alumni, students and innovators and anyone who's really interested in this subject to explore some of the key issues facing the sector today. There are many challenges, as we all know, facing agriculture and horticulture, and both Writtle University College and Liberty Produce are really pleased to organise this series to help start this interdisciplinary dialogue that we think is needed today. So just to introduce myself, I'm Anya Pereira. I'm Head of School of Sustainable Environments and Design at Writtle University College, and I will be facilitating this 1-hour session. I'm very, very pleased to be joined today by Dr. Dylan Banks, who's co-founder and knowledge director of Liberty Produce, and Dr. Susan McCallum, who is doing some important and groundbreaking research on blueberries for UK production at the James Hutton Institute. This is a rare opportunity to share the work of cutting-edge technology innovators and researchers with a wider audience and to explore issues from a practitioner point of view. So in these webinars, we are using the very broadest definition of agriculture to encompass both the horticulture and agriculture disciplines and their inherent connections. So without further ado, I'd like to pass to Dylan Banks to give you an insight into the innovative work that Liberty Produce are engaged with. So over to you, Dylan.
Thank you very much. Today, I'd like to talk to you about the combination of indoor and outdoor farming and how those combinations can allow for you as a farmer to produce more with less. Next slide, please. We at Liberty started our journey by building a research and development centre that we've co-located at the James Hutton Institute in Scotland, in Dundee. And in that facility, we have built a control system that allows for us to test at commercially relevant scales in terms of growing area and using the same technology and technology capabilities that operate at commercial scales, the various systems that one would need to grow indoor farming technologies. And ourselves and in collaboration with our partners, we've been working out how to address the various challenges that indoor growers, as well as protected crop farmers, as well as broadacre farmers face, and understand how our technologies can help them. Next slide, please. We've called our facility the Future Farming Hub, and the key thing that we have built is that we've been combining our control systems with lighting systems and nutrient monitoring and delivery systems, various hydroponic growing systems, sensor feedback capability, and all of these things combined allow for us to undertake both research and development that is strategically important for the sector, but also specifically important for both our customers and for our own internal product development roadmaps. Next slide, please. And here I'd like to play a short video.
Stephen McMenemy reports.
Could this be the answer to feeding ever-growing populations around the world? A new facility at the James Hutton Institute uses the latest technology to grow crops inside shipping containers. The work going on here is seen as vital to help the UK maintain crop requirements for the future. But it could also be used in countries where crops consistently fail.
This model, given that it's a container-based system, can be shipped anywhere around the world. It's not reliant on the natural environment at all. It's fully self-contained. We're controlling the environment within it.
This mode of farming looks set to be a growth industry. Stephen McMenemy, STV News.
Thank you very much. Next slide, please. And so moving on to some of the actual technological innovations that we've been able to bring together. The first of those is in the lighting space. Our approach in this industry was to look at the environment in respect of what growers needed, as well as in respect of what our competitors were providing from a lighting technology perspective. And in doing that, we were able to identify some significant gaps in that market that allowed for commercial opportunities that we've been able to develop products that can meet those needs. At the bottom of this, on the bottom right-hand side, you can see a picture of one of our lights. You can see that it's made out of an aluminium extrusion, and you can see that there's a little spider-looking blue cap at the end of there, and that spider blue cap is a RAS-approved water fitting that allows for us to cool those lights at the same time as shining the light onto the plants, which effectively allows for us to go at much brighter temperature, light intensities than we otherwise would be able to do because of the heat that's generated from the LEDs. This means that we can produce up to 350 micromoles at the canopy surface with a range of different colors and a range of different intensities without the difficulties of managing the heat that would otherwise be put into that environment. And this allows for us to produce greater mass of crops, fresh weight and dry weight yield, as well as improve other characteristics such as specific compounds or flavor or taste. Next slide, please. In addition to the lighting technologies, we are also developing nutrient control technologies. And in this regard, we're really interested in the first instance of understanding where the technology in the sector is moving, as well as looking to develop and build new technologies. And so what we did in this very first stage was to build a 12-channel nutrient control system, which is more than we think that we'd ever need in a commercial deployment, but allows for us to have that additional level of control that means that we can make the right decisions about which nutrient control is needed for which particular application. And this has been really interesting for us in terms of our ability to look at both new and novel research projects, as well as understand exactly the requirements that our customers have in this sector. In addition, we've been developing new technologies that allow for improvement of nutrient uptake within the plants. And these are specifically what we're calling micro nano bubbles, which are tiny little bubbles that we put into the water. And they're so small that they don't have the ability to push the water out of the way and float to the surface. And by doing that, we can ensure that the roots of the plants have got additional access to gases in that water. Could be air, could be oxygen, could be other gases too. And in doing those things, we've been able to show that we can boost the yield of those plants. Next slide, please. Lighting technology and nutrient technology are really fascinating, and they are complemented by our advanced sensor systems. And what we've been trying to do here is combine expertise and requirements that are traditionally seen within a research lab and a research environment, and understand where the exact expertise or the exact technological capabilities can be implemented within commercial scale systems, so that we can ultimately implement feedback control into our lighting and into our nutrients and into the other environmental control systems within the facility. And the combination of these things means that A, we can tune those plant environment characteristics to the betterment of yield or some other characteristic that you're looking to enhance within that plant. And additionally means that you can look to save costs. Perhaps with this kind of technology, you could reduce your electricity costs. And we are in the early stages of testing these feedback systems at the moment, but we are planning to bring our first products to market in 2022. Next slide, please. Yeah. And so examples of things that we grow in the Future Farming Hub are very wide and varied. We will be talking in a little bit about some of the excellent work that we've been able to do in collaboration with the James Hutton Institute. But just to show here, an area where we've had an early success and that we're really proud of is our ability to grow high yields of chives, and our ability to grow those chives with a vigor and a height that is suitable for particular applications in the commercial space. And in this regard, we've been able to achieve that with our technologies, where others of our competitors have not been able to meet that same requirement. And so that is one of the at the moment one of the leading areas where we've been able to show the power and capability of our technology, and we are hoping to be able to build upon those early successes and extend that into a range of other crops, which we're doing on a case-by-case basis, principally in collaboration with our key customers. Next slide, please. And so leading on to our collaboration with the James Hutton Institute and our development and understanding about some of the value that can be brought about from the combination of indoor farming with traditional farming technologies, our work with Dr. Susan McCullum, who will talk to you in much more detail about her research, has been extremely fruitful for us. We have ourselves been able to show that we've been able to reduce germination times quite significantly. Next slide, please. So by germinating within our facility, we've been able to reduce blueberry germination times from 10 weeks to 21 days. And this in of itself has led on to some commercial opportunities for us to develop and exploit our technology in this area that otherwise we wouldn't have known about without our collaboration with the James Hutton Institute, and we wouldn't have been able to test again without our collaboration with the James Hutton Institute. So key to what we've been able to achieve within the Future Farming Hub is engagement and collaboration, principally with the James Hutton at this point. But of course, we are keen to engage more broadly and work with others in the sector. Next slide, please. So just to sum up, the key elements that we've been able to demonstrate are that there is a place for combinations or synergy between indoor and outdoor farming, hybrid farming, where we can show that we've been able to improve the efficacy of propagation and reduce the time of propagation and the time of germination. We've been able to, and in that respect, this technology has got significant value in speed breeding programs. Additionally, our system that we've built is containerized, which means that it can work quite effectively in austere environments where the temperature and water requirements are such that it isn't possible to grow outside of the container, and we've been able to demonstrate that capability. Beyond that, the potential for this technology is quite significant. The key for us at this point, though, and the key that's been that we've maintained at every stage of our commercial development, is to ensure that the steps that we take, and we are ensuring that we take these developments on a step-by-step basis, have got material commercial advantage both for us and for our partners. Next slide, please. So the next area of development that we're moving on to is to take the expertise and the capability that we've been able to demonstrate at that commercial scale within the Future Farming Hub and shrink that down into a control system and into a growing unit that will allow for a greater number of trials to be done within a particular area and allow for wider collaboration with a greater number of research groups and customers. And at the moment, we're calling this product the Planter Vestium. It's a wardrobe that can grow crops, that can have the full climate control that's required to be able to grow those crops with the full control system that exists within the Future Farming Hub, that can have bespoke applications and designs and technological requirements as needed, but comes with the standard lighting system that is the same as that commercially relevant lighting system. So the product can be used as an early stage demonstrator for the capability of a scaled up system, but more importantly, the product can be used to optimize growing conditions and growing protocols to ensure maximum yield and maximum generation of other important compounds or organoleptic capabilities or aspects of those crops at a larger scale. And with that, I'd like to thank you very much for your time and pass you on to the excellent hands of Dr. Susan McCullum.
Thank you so much, Dylan. Thank you very much for doing that. And before we go on to Susan's, I know that some people joined a little bit late, so just in case you missed the first bit, we have got a question and answer session coming up at the end. So please use that function on your control panel on the right to ask your various questions. I can see that there are already some for Dylan, and we'll save those until the very end. But now may I just invite Susan to, because I think the two link very nicely together, to talk about her very interesting work that links the two projects together very nicely. Thank you.
Lovely, thank you very much. Thanks Dylan and Anya and Writtle University College for the opportunity to talk today about some of our research in collaboration with Liberty Produce. Our future direction is taking us very much towards controlled environmental agriculture, and this will allow us to look at speed breeding as a means to shorten the breeding cycle and increase genetic resources. So who we are a globally recognised research organisation delivering fundamental and applied science to drive the sustainable use of land and natural resources. So we're one of the largest research and development institutes for agricultural crops, land, water management across Europe. We have around 570 scientists on our site, and we are supported by about 165 PhD students. We are leading scientific innovation and plant breeding, and our main focus is on crops which are strategic to Scotland. So barley, potatoes, soft fruit, and that's looking at raspberries, blackberries, blackcurrants, gooseberries. Just recently, we've only really been doing blueberries in any real depth for the last 10 years. We've also brought in cherries, and we're looking at hops and brassicas too. So where are we then? We're based across a number of sites. We have two main research sites, one which is up in Aberdeen looking at landscape, soil and water conservation, and they also have a lot of scientists studying climate change. I'm based in Invergowrie, just outside Dundee, and we look at a lot of breeding and genetic resources. We have huge capabilities of land and laboratory space, as well as glasshouses and some controlled environments. We have two main farms as well. We have Balrudley, which is just around Angus, and it has about 170 hectares of arable farm. And we also have Glensall, which has been in our hands since 1943, looking at a lot of land research. And this has been really useful in the soft fruit because an awful lot of native blaeberries and lingonberries can be found up in Glensall. So it's really useful for our genetic diversity studies. So what we do then, we have a vision to be at the forefront of innovation and transformative science, and that's looking for sustainable management of land, crops, natural resources that support thriving communities. A lot of the work we do up in Aberdeen is an awful lot of data looking at how we live now, how things have changed, and especially living through the pandemic, how that's changed how farmers work, how scientists work, how the community go about purchasing and getting hold of any of the amenities of shopping, because it has changed our shopping habits quite a lot. We also do a lot of work with soil because we can't grow crops without having the appropriate soil, and that's looking at the physics, the chemistry, the biology. We need water, be that for drinking or for the quantity or quality that we need for watering our crops, and we have to make sure that that's feeding into what the communities around us are actually looking for. We also do a lot of DNA fingerprinting, so a lot of growers, if they're suspicious or they're concerned about some of the varieties that they're growing, they can take leaf material and send that to us, and we can fingerprint that to see that it's true to type, that it is that they're growing the selections that they think they're growing. We hold seed banks of national importance, in particular potato seed banks. We have a soil archive. We have approximately 43,000 samples that have been collected across Scotland, and they've been collected since, again, the 1940s. And we also look at key links between biodiversity and ecosystem functions. So we try and look at the whole picture. We're not just looking to improve crops or grow crops. We're looking to make sure that it's sustainable, that we're looking after the soil, the land, and the communities that we serve. So in terms of the plant breeding side of things, which is where my real interest comes into, we're world-leading research for potato, barley and soft fruit breeding. Currently, potatoes, it's at a small scale, 5% of the UK market share, but we do have our own potato breeding company, and we do a lot of potato research with other private breeding companies. Black currants, I think we are responsible for 99% of the varieties that are grown in the UK, 50% of global market share. We work very closely with Suntory, who now take over Ribena. So most of our selections for blackcurrant are grown specifically for Suntory and for their needs in terms of the antioxidant capacity, how much juice there is, vitamin C content. Raspberries, we have 70% of the UK raspberries are grown by ourselves. So if you come across any of the Bens, that's our blackcurrant. So we've got Ben Nevis and we've got Ben Ample. So there's a lot of the Bens. For as far as the blackcurrants, they were bred at the James Hutton Institute. For the raspberries, it's all the Glens. So there's Glen Moy, there's Glen Lion, there's a recent variety that was released called Glen D. So any of the Glens came from our institute. Blackberries, we have the #1 variety in Europe, that was Loch Ness. So again, look out for any more lochs. And we have a new blackberry breeding programme now. So hopefully we'll be able to start to get some new varieties out on the shelves, and they'll all be preceded by lochs. Sweets, we have 40% of the market share in the sweet breeding. And blueberries, our breeding programme just launched in 2017. So we're not quite ready to release a new blueberry yet, but we do have some fantastic selections that are coming soon. So hopefully we'll be able to get back to you at some point and get you to look out for our new blueberry selections. So the breeding stages then, conventional breeding takes between 10 and 15 years in a perennial crop like blueberry. So that's from the time that we do our first crossing to germinating the seeds, to growing the seedlings, to getting them into field trials where they're evaluated year after year. We can't just evaluate them in 1 year because a lot of the traits that we're interested in, which is fruit quality, fruit size, the Brix and the acid, the taste, the flavour, a lot of that is very environmentally dependent. So we have to grow it over a number of years, and we also try and grow in a number of different places. So in the blueberry breeding programme, we have partners that are all across the continent. So hopefully when we get a selection ready, we'll have it trialled all over the place to make sure that we're not just releasing a variety that's perfect for Dundee. We need to know that it'll stand up if it's grown any place in the UK or Spain or Portugal or any place else. The UK market is still worth in excess of £400 million for blueberries, and there's no signs of slowing down at all. So we know that investing in blueberry is something that's really important, and it's something that we really need to focus on and do it a little bit more. The appetite in the market for UK soft fruit is evident. But what we need to do is we need to speed things up. Conventionally, 10 to 15 years is an incredibly long time between selections and actually getting something that's ready to go into market. So anything we can do to shorten that through innovation, which is what we've been doing with Liberty Produce, and through molecular markers and understanding the genetics, will definitely pave the way for an awful lot quicker selection protocol and cost savings as well. So what we've been trying to do with our initial research with Liberty is how to break the annual cycle. Dylan showed a very nice picture in terms of the germination. And under current circumstances, when we do our breeding in the glasshouse, we do our crosses, and then it takes a long time for the fruit to develop, then we extract the seed, we germinate the seed. We can only ever do one of these things once a year. But working in a controlled environment, what we can potentially do is take things out of season. So we can do crosses and germination. Instead of once a year, we can do it twice a year. If we can speed it up even more and have plenty of room, we can potentially do it 3 times a year. And in order to meet the challenge of feeding a growing population, breeders and scientists have to find new ways to increase this genetic gain in the crop development. And the more generations you can do, and the more you can bring in diverse populations and look at novel or the plants that we're finding native, then the more you can increase this diversity. So what we've been doing essentially comes to what we call speed breeding. So the advantage is obviously time. We're cutting down the time that it takes to develop new crops. We're cutting down the costs, because one of the biggest costs within breeding programs is all the selection times. It's putting it into the field and it's analysing it year on year. We can bring in more diverse germplasm, native crops, look for resilience. An awful lot of the seedlings that we take out to the field, they don't survive. And we know they won't survive for various reasons. They don't have the root system that they need. They don't have the vigour that they need. In speed breeding, we can start to screen that within a controlled environment. So we only take out the seedlings that we know are strong enough and resilient enough to cope. And that's where some of the markers come in as well. We can screen that to make sure that it's got the genetics that we're interested in. It gives us environmental control. As I mentioned in our breeding programme, we have partners across 3 different continents. So we can set that to make sure that the crops that we're selecting for them are likely to hold up in their own native environment. And it's collaborative research, which we just can't get enough of. We know that on our own we can only produce so much, but putting our net out wider, working with innovation and technology producers like Liberty, it lets us get this research, it lets us get there quicker. If we're able to shorten the length of time to develop new generations and increase the levels of diversity within the breeding programme, we can better screen the plants for resilience before we even put them into the field. And that's where our goal is with the speed breeding. Now, Dylan's already mentioned before about the light system that they have, and this is just a very picture that I've got off the internet that just shows the different wavelengths and how they react differently to the plants. So, you know, a bluer light will enhance the chloroplast activity, promote cell division, so plant growth. Same with the red light, it also stimulates germination and flowering, and then you've got more of a purple light that covers everything that's going to be suitable for all plant growth. If we can extend the duration a plant is exposed to light each day, or target the specific wavelengths to promote flowering or vegetative growth, it becomes possible to reduce the generation times under favourable conditions. So some of the groundbreaking research which has paved the way for the speed breeding. Speed breeding is not new in any form at the moment, but it's new to soft fruit. Nobody's doing this in soft fruit at the moment. NASA's been doing it up in space for the last 20 odd years. The Hickey group in Australia, they started an awful lot of this working with John Innes, and they were looking at wheat and barley and oats, brassica, and they've made great progress. I mean, they've been able to do generations not just 3 times a year, and some of these crops up to 6 times a year. So they really have developed and enhanced their breeding programs for a number of different programs. So that's something that we really want to try and do within soft fruit. We're not aiming for 6 generations just yet, but if we could even double it to 2 generations, we're making huge leaps on this genetic gain. So the next couple of slides, these are just a few of the pictures of our protocols that we've developed with Liberty Produce. So this is a snow chaser blueberry that went into Liberty Produce last year, and they were able to germinate it so much quicker than us, 3 weeks instead of 10 weeks. And they've looked at different ways of growing them hydroponically to develop the above ground shoot length as well as the below ground roots. So we've been doing a lot of studies now just to see how we can tweak the blueberry growth stages that little bit quicker, and making sure that we give the crops the right nutrients, which again, Dylan's already shown how much they can adapt that, making sure we get the good root growth and the good above ground. So hopefully when we then take that out to the field, we know that it's going to be a resilient crop and it's going to withstand the Scottish summers if we get such a thing. And this next slide, this is just again some of the snow chasers that's come out. We've brought it out of the hydroponics and we've put it into the substrate, and in a matter of a week, it's come on really well, which just shows how strong the crops are when they first came out of Liberty Produce. So the facility is definitely working, and hopefully we can enhance that as we go forward. So our overall aims then, it's all about designer crops and biodiversity. We need to make sure that we're developing crops for a particular environment, and we can't do that without using a controlled environment. We're looking for increasing this biodiversity for food production, sustainability, waste reduction, and being able to grow crops outside the natural environment and reduce the timescales. And this is really important. I think Dylan, again on his slide, had about Africa. There's a lot of work going on in Africa to try and develop crops that's specific, that we know can then grow within their environment. So many of the modern cultivars that we have now, they were bred for a resource-intensive cultivation system. They have a very limited environmental tolerance and a narrow genetic base. So we know that something that produces and grows really well in Florida, we bring it to Scotland and it wilts and dies, it does nothing. So we need to know that when we're looking at stuff, that it has that resilience and it has that environmental plasticity. We've already seen devastating consequences seen with a lack of diversity. The banana, which we all know and love, there are thousands of species of banana of all shapes and sizes, but more than half of the global production is just one species, and that's called the Cavendish. Back in the 1950s, the banana of choice was the Gros Michel, and it was wiped out by Panama disease, a fusarial wilt, and it led to the Cavendish becoming its replacement. But at the time, the Cavendish was thought as smaller, it was less tasty, but it could be grown in the ground that had this fusarial wilt in it. And that's why we were then able to use that as a replacement. But it was because there wasn't a second choice. For most food production, once you've got one crop that's a really good yielder and consumers like, you focus on that and you put all your money and effort into that. And sometimes the breeding side of things and the new species comes to a stop. Currently, there's a new strain of this Panama wilt, which is deadlier than the first strains, and it's destroyed in the last 5 years more than 10,000 hectares. And so the race is now on to find halt the progression or find an alternative. And that's why it's important that we keep biodiversity in the forefront of our mind. We can't just rely on one species or one crop, because if something comes along, and given enough time, something will come along and start to halt that progression or wipe out the crop altogether. I think there's a corn blight in the 1970s that cost, I think it was 70 or 80% of the corn was completely wiped out. It cost over $1 billion because there was nothing to replace it. Everything had been put onto this one strain. So I think it's really important that we are able to bring diversity into our germplasm and make sure that we are sustaining, not just for today, but for the future. So what the future holds then for James Hutton Institute and Liberty Produce is hopefully developing new protocols for blueberry, raspberry and blackcurrant in order to develop this speed breeding. If we can adapt and perfect the light and the temperature regimes, we can increase the generation rates. And by increasing the generation rates, we're increasing diversity. For perennial crops like soft fruit, the major driver determining the breeding cycle is the level of juvenility. So that's something that it makes it a bit trickier than the barley and the wheat that's been developed before. Juvenility is this extended period post-germination, which is a vegetative development in which flowering is repressed. So even under favourable conditions, the plant will not produce flowers. And the reason for that is the plant wants to make sure that the root system and the above ground architecture is strong enough and has enough photosynthetic capacity to be able to maintain that plant. What it doesn't want to do is produce flowers in year one and then it starves itself of everything else, because it's a huge carbon cost to the plant to produce flowers. So it makes sure that it does not allow it to happen in the first few years in order to make sure that the plant is healthy and it's sustainable. There are various lengths of time for that as well. In blueberry, it's normally around 2 years between your first cross, your seedling, and the juvenility break-in. Berries, it's around 3 to 5 years. Avocado, it's up to 15 years. So you can see that this juvenility period is a big thing, but I think if you're varying the right light wavelengths and the right temperature at the right time, we can probably bring that down just a little bit more. The varying degrees of critical day length across the soft fruit has to be considered with the photo period adjusted accordingly for successful gain in this generation rate. The good news is the initial work that we've done with limited produce has indicated that this will be feasible with soft fruit. So that's what we're working on now, and hopefully we'll have protocols out in the very near future. And at that, I would like to just thank you for your time, apologise again for my minor technical issues, and just thanks to Writtle University College again, Limited Produce, and our partners within the breeding programme. Thank you very much.
Thank you, Susan. That's really interesting, and thank you for doing a sterling job of carrying on when the technology fails. That's brilliant. So if I could just sort of summarise what you were just saying, so I think what your work is basically showing is that that accelerated germination allows successive generations over a shorter period, and you were saying about how that will shorten potentially a breeding programme. But does it fit into a conventional breeding programme then? Is that what happens?
Oh, absolutely. I mean, at the moment, we're doing it purely for the scientific base of things, just to see how much we can manipulate and speed up the germination time, and in order to manipulate the juvenile period. If we can start to break that without damage to the plant, and that's important, because there has been some studies where they've taken a crop and they've given it light, for 23.5 hours a day. And it will respond differently, and it will produce flowers, and it will produce fruit under these conditions, but it's not sustainable for the plant. So it's important that when we look at any of the protocols that we develop, we need to know that we're not harming the plant in this process. We want to speed things up so we can cross quicker and we can get that diversity up, but ultimately we want to then be able to take that plant back out into its native environment, and it thrives and it produces a really good yield and a good crop.
And sort of a follow-up question to that is, have you found any difficulties about taking it from a hydroponic system to put it into a conventional growing system at all? Or is it dependent on variety that you're using?
Yeah, I think variety will play a very big part in it. I mean, a lot of the blueberries, there are different species of blueberries, and there are a lot of very different varieties, and they will perform differently. Even when it comes to just normal propagation or germination rates, the variety in the species does play a big factor. So genetics is a big part of that. But so far, the species and the varieties that we've grown from Liberty and then taken back out to plant have performed exceptionally well.
Okay, thank you. So if I could just call Dylan back in, because I think there was a question specifically for Dylan about the nano bubbles and biostimulants, and whether there's been any interaction between that. Has it increased the efficacy, for example, of biostimulants? Have you been able to look at that?
Yeah, that's a really interesting area of research, and anyone that is looking at that area, I advise them that's the right direction of travel. Certainly. There is a growing body of evidence, both in the literature and within commercial scale applications, that the combinations of those things are interacting positively. Again, because of the commercial nature of the actual work that we're doing at this point, I'm unfortunately not in a position to talk about the exact bits of work that we're doing, but yeah, that's that particular viewer has really identified a key point there.
Okay, so I mean, I think one of the things that we want to just talk about a little bit, I think, is this relationship between what Dylan's work is doing with Susan's work as well. But I wanted to ask you a general question to both of you. In terms, obviously, we've spoken a great deal about blueberries here, which I think most people wouldn't have thought of as being something that would be suitable for the system, but obviously it is. So from your work and your experience, do you think that this could work with any crop, or are there limitations?
You perhaps it's it was our naivety, but I have to say we didn't expect to see such beneficial outputs as well ourselves, and so we've really lent into this and are now exploring the value where this technology can add in a much broader range of crops. I certainly, what we've learned through this process is that we needed to be much more open-minded about where the value proposition of the technology and the technology development could be. And again, the answers to those questions, which may not be apparent on day one, are, for us at least, have been brought about through collaboration. And so that's certainly we will be hoping to explore other areas of value where we'll be able to work with Susan and her team and the James Hutton in understanding the value that the target crops that they're working with in the first instance, where we can add value into each of those areas.
One very practical question for you, Dylan. In terms of the containers, do you actually have a set idea of where they could be positioned? Do you think they could sit on an individual farm? Could they be in a sort of agri co-op? Is there any limitations to where the bigger containers can be situated?
So yeah, again, really interesting question. Each business model needs to be looked at in its own right, and the strategy that we take internally as a business is to make sure that we understand what our customers are trying to achieve in that respect, and you configure the growing system to meet the needs of what that customer is trying to achieve. So certainly some of our customers do look at on-site farm deployment, and again, actually, where we are engaging on a cooperative basis, the idea is that depending on what you're looking to produce and in terms of what the value is, then a collective ownership or positioning can also have work equally as valuable.
Okay.
Sorry.
No, carry on.
Last thing we're looking to look at is multi-purpose, multi-use. Perhaps in one period of the year, the indoor growing system can be used to produce one thing, and then one type of crop, and then at another point in the year you might look to utilize it to do something else. And in that way, identifying where it is complementary and can provide value to broadacre and other growing technologies, those are really the areas that we're looking to try to find areas where we can add most value.
Thank you both very much. That's a very fascinating story to be told about how the innovation and the technological research has fitted into a research program. I think that's it just showcases that when we all sit together and look at a problem, we can actually solve it if we think in a very integrated way. So I do thank you very much. So our hour is up. It's gone really quickly. Thank you everyone for bearing with us with the technical problems, but I think it was a very interesting hour. Just like to say a huge thank you to Dylan and Susan for explaining their work, and also to those who are working behind the scenes in promoting today's event and the technology. And I think we've had a really interesting conversation. I hope we've answered most of your questions, but if we haven't, please contact us and we'll happily get back to you. And there will be a link to the actual webinar sent out shortly. So I think one of the things that's come across to me is that when we come together, we learn a lot. I've certainly learned a lot today, and thank you very much, and I hope you've enjoyed it as much as I have, and thank you to our audience. So to our audience, please join us on our next webinar, which is on the 24th of February. Thank you all once again. We will leave you with a very short video, but until next time, we wish you a very pleasant evening.
The great outdoors, and even greater indoors. An education for the real world. Putting theory into practice. Smaller class numbers and supportive tutors. Highest overall for student satisfaction. Breaking new ground. Breaking the mould for almost 130 years. Around 200 open hectares outside Chelmsford. A close community that will welcome you inside. Making lifelong friends. Experiences for life. Being just 35 minutes from London, but a world away. Discover our hands-on, industry-recognised courses. Putting theory to the test. Putting alumni in high places. Putting your career first. Putting science into practice. Writtle University College.